All right, let's get started. Hello everyone, welcome to our workshop today. My name is Sydney Thomas. Before we begin, I'd like to let everyone know that this workshop is being recorded and we will be providing you with a copy of the recording after the workshop is complete. Also, if you have any questions at any point in the workshop or you get lost at any point in our tutorial, please do put that in the question and answer box below. My colleague Joe and former colleague Helena will be monitoring that box and we'll get you back on track as soon as possible. So before we dive straight into analysis, the first thing we're going to do is quickly go over what molecular networking is and how it can improve your untargeted metabolomics analysis. So if you're here, I hope you would agree that untargeted metabolomics can be an incredibly powerful discovery tool. A single untargeted metabolomics experiment can identify tens of thousands of metabolites in a sample, yet actually figuring out what those metabolites are, a process we call metabolite annotation, can be very difficult and is often the biggest hurdle to really using this data well. So let's say you have an experiment where you're going to do untargeted metabolomics to find all of the possible metabolites of the drug azithromycin. The first thing you're going to do is you're going to collect a bunch of samples under different conditions. You're then going to put those samples on your mass spectrometer, and that mass spectrometer will provide you with an MS1 spectrum with the mass and retention time of all the metabolites in your sample. Now, this MS1 spectrum can tell you the chemical formula of your metabolite. It can also give you the relative abundance of that metabolite between samples, and it provides the retention time. And while all of this is very important, in most cases, with just this information, you're not going to be able to really identify a metabolite with confidence. That's because many metabolites have exactly the same mass, even though they have very different structures and functions. So in untargeted metabolomics, we perform a second round of mass spectrometry called MS2. But in this round, instead of measuring the mass of the entire metabolite, we break that metabolite up into bits, which we call fragments, and we measure the mass of the fragments. And what this provides is information on the chemical substructures within your metabolite. And you can see how if you get enough of these fragments, you can start to piece together the structure of the entire metabolite and identify it with a lot more confidence. Here's an example of how this works in real life. Well, actually, the practical way that we do this is through a process called library annotation. So in library annotation, you're going to take an experimental spectrum. So in this case, this is the MS2 spectrum of azithromycin. And you're going to compare it to library spectra, which are MS2 spectra from known compounds. And you're going to use an algorithm that compares both the mass of your MS2 peaks as well as their intensities to find the library spectrum that is the best match to your experimental spectrum. So here is an example of how this works in real life. Here at the top, we have the experimental spectrum for the molecule sucrose, and you can see it is an excellent match for the library spectrum for sucrose. In this case, the algorithm that we're using to do the matching is called a cosine score. A cosine score of 1 is the best and 0 is the worst. And so you can see at the top here, we have a perfect match, so our cosine score is going to be 1. However, if we then take that experimental spectrum for sucrose and try to match it to a different library spectrum, say the library spectrum for azithromycin, you'll see that that match is not very good at all, and so our cosine score is going to be close to 0. Now, this process works really well if you have an excellent MS2 library that contains every molecule you would ever want to see collected under the exact conditions that you're doing your experiment. However, what happens when you're looking for a novel metabolite? Or say you're working in an organism that's not very well studied? What happens when there are no matches? And this is actually not a niche use case. Only about 10 to 20% of metabolites are annotated using library matching using our best-in-class publicly available libraries. Now, if you have a really excellent private library and you're working in human serum, maybe you can get that number up to say 30 or 40%, but that still means that you're throwing out the majority of your metabolites, which remember are your drug candidates and your natural products, just because you have no idea what they are. And so this is where molecular networking comes in. In molecular networking, instead of just performing that library annotation step where you compare experimental spectra to library spectra, we're going to perform an additional step where we compare experimental spectra to each other. And in this case, we're not actually looking for exact matches, we're looking for close matches. And that's because since MS2 gives us information about the chemical substructures within a molecule, chemicals that have similar MS2 spectra tend to have similar structures as well. And so what this ends up looking like is that all of the metabolites in your experiment will be represented as a circle or a node. And if the MS2 spectra of two metabolites are similar enough, we're going to draw an edge between those nodes. Now, if you apply this to an entire experiment, you get a molecular network. Molecular networks are organized into subnetworks, which tend to represent specific molecular families. So you'll often see, say, a subnetwork for lipids or amino acids or a specific class of natural product. And once you dive into these subnetworks, you'll see that some of the metabolites in the subnetwork have library matches and some do not. But now for those unknown metabolites, you have a list of known metabolites that are structurally related to your unknown metabolite of interest. Here's an example of how this works. Here in this subnetwork, all of the metabolites with library annotations are all different bile acids. And so it's not much of a leap to hypothesize that your unknown metabolite is also going to be a bile acid. So the next thing we're going to do is to dive in and try this ourselves. But before we do that, I'm going to give you a little bit of background information about the platform we're going to be using today. Today we'll be on the demo server for Ometa Labs. Ometa Labs is the commercial version of GNPS, which is now GNPS2. GNPS was the original molecular networking platform, but now it includes an entire suite of platform-independent tools for untargeted metabolomics analysis. Since GNPS was designed for academics, it's designed for sharing, collaboration, open science, and public data. All of the data that is run on GNPS is housed on a shared server and is run using shared compute. There is a lot of support provided for GNPS users through online workshops like this one, as well as a lot of other online workshops as well. Actually, Helena, could you post those links to the other GNPS workshops in the chat when you get a chance? There are a ton of resources available to you, including resources that are not just in English, which is really cool. In general, GNPS is developed by the GNPS2 team. If you have a tool that you want to create, you can collaborate with them to develop it. But in general, they're going to be trying to create new tools that will support kind of the wider and broader academic community. So one of the great things about GNPS is that it is free for all academic and nonprofit researchers. But it is neither GNPS nor GNPS2 has ever been available to commercial entities. So that's where we come in as Ometa Labs. We have a platform that is very similar to GNPS. So if you're an academic in this workshop, don't worry, the stuff that we'll be showing to you today is still applicable if you want to use GNPS. However, we have designed our platform for more secure sharing and collaboration. We've enhanced the data privacy and security. We do allow for on-premise deployment and provide dedicated compute for all of our clients. In addition, we do a lot of one-on-one support and consultation, and we can do custom development that's tailored to your organization. So if you have a niche use case, we can make a workflow that works for that. For all of this, we do charge an annual fee, a good portion of which is actually kicked right back to the GNPS2 team so that they can keep creating new awesome untargeted metabolomics tools. And while we do mostly work with businesses, if you are one of those academic or nonprofit researchers who say for some legal reason can't make your data public, you can come to us and we can help you out with that. So that's it for the platform. Let's get to the fun part of the workshop, which is trying it ourselves. So I'm going to have you guys navigate to our demo server. I'm going to paste this information in the chat so it's a little bit easier to just copy paste. So you should see that link in the chat. That will get you to our demo server. Once you're on our demo server, if you look in the top right-hand corner, you can use this username and password to log in. This should be fairly obvious, but this is our public-facing demo server, so please don't try to, say, upload any of your own data to this server or do anything that you generally wouldn't want everyone else on this workshop to be able to see. We're also definitely breaking the record for how many people are trying to access this server at once today. So if something takes a while to load, apologies in advance. You will have access to the server after the end of this workshop. So if you want to come back in a couple days and go through this yourself at a time when not everybody else is trying to do it at the same time, you are always welcome to do that as well. So I've put this information in the chat, so you will still have it. I'm going to navigate back to the Ometa homepage. Hopefully you guys can all see this correctly. Once you log in, you will see this landing page, which is where you can access all of the tools that Ometa Flow has to offer. The first thing I'm going to point you towards, though, is the file browser, which is right here. Actually, if you click on this right now, since you are a viewer, you won't be able to see it, but I'll show you what it would look like if you are not a viewer. It looks like this. So this is your own personal private browser where you can upload all of your mass spectrometry data. You do not need an FTP client to upload data to this. You can just drag and drop or use the upload button here. You can also rename, delete, create folders, and organize your data just like you would in a normal file browser. Now you'll see here, I've already uploaded some data for us to analyze today. If you click into this, you'll see that we have a couple folders containing mass spectrometry files. These mass spectrometry files are MS data that is in MZML format. So MZML is an open source format that allows us to use data from any instrument vendor. However, if you get a 502 forbidden error, that's just because your user is a viewer right now, and so you can't actually see the file browser. You will be able to see everything else. We're just trying to make sure that people don't accidentally upload their data to this server because it's not very secure since it's our public server. So anyways, the MZML file format is open. It's great. We wish all of the instrument vendors used it, but unfortunately, they all have their own proprietary formats that they prefer to use. And so you will likely have to convert your files from those proprietary formats to MZML. There are free tools that allow you to do this. Unfortunately, we are not legally allowed to include these tools on our platform. So if you have questions about this, there are a lot of resources to help you convert your files, or you can just come to us and ask us what's going on, because unfortunately it's more work than we wish it was. So this is the file browser. It is private to you. It is where you can put all of your files. But if you say have a data set where you know everyone in your organization is going to want to access it, or if you're on GNPS, I guess everyone in GNPS would want to access it, you can upload it to the shared browser here. It looks exactly the same as your file browser, except that it is accessible to everybody on your server. So now you know where your data lives, we can start to analyze it. So let's go. So in Ometa Flow, all of the tools are called workflows, and you can access them by clicking on the workflows button here or the launch workflows button here. So once you click on that, you will pull up all of the workflows that are currently available on Ometa Flow. We have about, 53 right now. Some of these workflows are definitely a lot more popular than others, and I would say our most popular workflow is this classic networking workflow right here. So to get that started, you can just click the Launch Workflow button under Classic Networking. And this will pull up the workflow input page that allows you to set all of the parameters for your analysis. Sorry, one second. I forgot that if you join a little late, you won't see the chat. So I'm going to put the link back in the chat for everyone. If you did not get that link, let me know. So this should be the link that you can get to get onto our demo server, as well as get your username and password. All right, so here we have the workflow input for classic networking. The first thing you're going to do is enter a job description here. This can be anything, it does not need to be unique. And then we'll scroll down to our file selection options, where you can choose which files you want to analyze. So you can see here, we can either manually separate your data into up to 6 different groups, or you can always add a metadata file here that allows you to separate your data into as many groups as you want. To select your data, all you're going to do is click on one of these green buttons here. This is going to pull up your window, which allows you to select files. So you'll see here we have our personal file browser with your username, but we also have the shared browser if you want to access anything there. For today, we have some samples that are either process blanks or samples from people with omnivore or vegan diets. If you look in here, you can see we have our MZML files right in there. So to add samples, you can either just click on them individually, or you can click on the folder that they're in. So for today, I'm going to add all of the blank samples to group 1 by clicking here, and then clicking Add Selected File to Input Data Folder Group 1. Then I'll add my omnivore files to group 2 by clicking here, and then clicking on group 2. And my vegan files to group 3. Now you'll notice I also have a metadata file. So I'm going to click on that and add it to my metadata by clicking Add Selected File to Metadata Path. And now we're going to close this window and see our selections. So you can see we have our blank files in group 1, our omnivore files in group 2, and our vegan files in group 3. You'll also notice that our metadata file is right here as well. If you've selected anything wrong, you can also always click on this Remove File Selection button here and remove anything that you didn't mean to click on. Now, the one other thing that you will probably be doing on a regular basis is choosing which spectral libraries you want to use for library annotation. By default, Ometa Flow offers all of the publicly available libraries from GNPS, MassBank, and Mona. And all of these libraries are included in this LC folder here. But if you have a specific library, then you only want to use that library. You can always click Select Spectral Library folder here. Go to the Libraries tab right here, and you'll see here is our LC folder with all of these different libraries in it. You can click on any of these libraries and then click Add Selected File to Spectral Library folder. Close this window, and you'll see now we've selected that specific library. Now, if you ever do this, remember to remove the file selection for the overall folder so that you're actually just looking at this library and not this library plus all of the other libraries in that folder. So this is it for file selection. If you keep scrolling down, you'll notice we have a lot of options for this workflow. I promise this is more complicated than most of the workflows that we run. But thankfully, we have set the default values to be just generally good for most of your everyday analyses. So in most cases, you're probably not going to change much in here. If you do have questions about what any of these parameters mean, you can always hover over the tooltips here to get a little bit more information on the parameter, or you can go to our documentation right here, which provides you with a lot more information about all of the workflow inputs, all of the outputs, as well as some tips and tricks about how to run the workflow. The one thing we are going to change today is if you scroll almost all the way down to the bottom, you'll see this section for Advanced MS1 Peak Integration. This is actually a Ometa Flow specific feature. So if you are familiar with GNPS terminology, this is a feature that allows you to do feature-based molecular networking within a classical molecular networking run. So it will perform peak integration while it's performing molecular networking. If you are not familiar with GNPS terminology, don't worry about it. It's just a cool tool that allows you to calculate peak areas while you're doing the networking task. So to do the peak integration, all you have to do is click on this from no to yes, and that will perform your peak integration for you. Now, usually what you would do once you've got all of your parameters that you in place the way you like them would be to click on this submit workflow button here. But for today, instead of submitting a workflow and just sitting around watching it run, we've already run some of these tasks for you. So instead of clicking submit workflow, you're going to go to your tasks up here. So if you can click on this tasks button, you'll see all of the tasks that have been run by your user on this server. So for now, we're going to go over to our classic networking task, and we're going to click view task. This will pull up the task status page for molecular networking. You can see we have a lot of results down here for you to check out, and feel free to click through on your own time. For today, I'm going to highlight a couple of things. First of all, we have our logs, which tell you how the workflow ran and how well it completed, also how much time it took. We have an option to clone a task. So say you ran a task and you want to change one parameter, but you don't want to go back and change every little thing that you had already changed. You can clone this task and it will create an entirely new task for you with all of the original parameters from this molecular networking run. You can also hide tasks, delete tasks, or protect or public tasks. Making a task public allows everyone on your server to be able to see a task. Usually most tasks that you run are only visible to you and your systems administrator. If you want to share a task, this is an easy way to do it, but you can always just copy and paste this URL as well. And anybody with access to your server can just check out these results from their browser, which is really nice for collaboration. If you don't want people to be able to see a task using this URL, you can always click on this protect task button and it will make the task only visible to you, and nobody with this URL will be able to access it except for you. Now, usually this is a platform that's designed for collaboration, so we don't use this button that much, but if you have something that's really important to protect, you can always click on it. Now, you see we have a lot of results down here, but this is actually not all of the results that are created by this analysis. To get to those, you can click on the Browse All Results button here, and that will pull up a file browser with every single file, even the intermediate files that were created by this analysis. Now, there are a lot of files in here, but just for future reference, there's one file I like a lot, and it's this jobparameters.yaml file. This file has all of the input parameters that were used to run this workflow. So if you are somebody who uses electronic lab notebooks, this is a really nice little file to just take, download, and upload to your electronic lab notebook so you know exactly how you ran this analysis and you can always reproduce it in the future. So there's a lot to look at here, but for today, we're just going to go right to our molecular network, which you can find by clicking this Visualize Full Network in Browser button here. Because a bunch of people are trying to do it at once, it may take a little while to load, so I've already loaded it. Apologies if it takes a little while to load for you. But once it does load, what you'll see is this molecular network here, where we have larger molecular network that's split up into smaller subnetworks. Now you can zoom in with your mouse and you can look at any of these subnetworks. And if you click on one of these circles, which are each of your metabolites, you can see that we are pulling up your MS2 spectrum for that metabolite here. If the metabolite has a library match, the circle will be blue. If the metabolite does not have a library match, the circle will be gray. So you can see this particular metabolite has a library match, so we can see what the library match is, as well as the structure of that metabolite if that was provided in the library. If you want to compare two metabolites, you can always click on the edge between nodes, and what you'll see is the MS2 spectra for both of the connected metabolites. And if you click down here, you'll also see a mirror plot that compares the two metabolites. Now, if you ever want to see this mirror plot in a larger format, you can always click this View Mirror USI button here. And once that loads, you'll see the mirror plot, you'll see the spectrum similarity between those two, as well as the list of MS2 peaks for each of your two spectrum. So there are a lot of really cool ways to kind of make these personalize and customize these networks. You can feel free to try them all out on your own time. For today, I'm going to show you a couple of cool things. The first of which is the ability to draw pie charts on your network nodes using your group information. So if you scroll down here, you can see we have this pie configuration section here. This allows you to draw pies using your input groups. So here we have the input groups that we manually created, but you can also check out any of the input groups that you added in your metadata. So one of the input groups in my metadata was diet. So I'm just going to click on that, and you can see I have my blank groups, my omnivore groups, and my vegan groups. To draw pie charts, all you do is you click yes here on this show pie charts option. And if you scroll back up here, you will see we have now drawn pie charts on our molecular network. So this is really helpful if you're doing something that has blanks, because we now know our blanks are pink. So you probably don't want to follow up on, say, metabolite 238, because it was only found in the blanks and not in any of your samples. Now, I'm going to actually turn these back off, and we're going to look at a specific network component. So to do that, you're going to scroll down to the network controls right here, which is right next to that pie configuration option. And you're going to enter the number 13 into the component filter. Now, you will probably have to scroll out and scroll back in to find this component. But this component is actually that same component that I was showing you in the PowerPoint at the beginning of the workshop. So this is a component in which all of the library annotations are to bile acids. And so we're going to hypothesize that some of our other metabolites, unknown metabolites here, are also going to be bile acids. Now, if you draw those pie charts again, you'll see that some of these bile acids are found in both omnivores and vegans, but some of them seem to be specific to omnivores or vegans. And so while this gives us a quick idea of some of the metabolites that might be specific, we probably want to get a little bit more of a robust statistical analysis to see which metabolites are which of these potential bile acids are upregulated in one diet versus the other. And so to do that, we have a statistics processing workflow that's built into the platform. So if you go back to your molecular networking task, which you can always do by clicking the back button twice or by clicking on this tasks button right here, you'll see we have a bunch of options for downstream analysis. And for that, we have a bunch of options, but statistical analysis is the one we're going to be doing today. You'll notice it mentions that it required MS1 peak integration. That's why we changed that one option that we changed in the workflow input, because that provided our MS1 peak integration information that we're going to use to do our statistical analysis. So if we click on this button, we will pull up the workflow input page for statistics processing. You'll see that we've already selected all of the correct files for you, so you don't have to go back and figure out which of the files you need to use for this task. And now we actually only need to add in about 5 different options here instead of like the 20 that you have in classic networking. So the first thing you're going to do is tell the workflow which column in your metadata file contains the information that separates all of your samples into groups. Mine was called diet, so I'm just going to put that in. We also have the ability to filter metabolites that have a large number of missing values. So in this case, the default is to get rid of any metabolites that aren't present in at least 50% of your samples. But if you have different use cases, you might want to change this number to 100%, or you might want to change this number to 1%. So that is entirely up to you. We also have the option to perform blank subtraction. So if you have blanks in your data, like I do in mine, you can get rid of any of the metabolites that are found in the blanks and high concentrations. If you want to perform blank subtraction, You do have to tell the workflow which column in your metadata tells you which samples are samples and which samples are blanks. In this case, it's the same column for me. I had my sample groups and my blanks in a single column, so I can just use that. And then we have the option to perform machine learning analysis. So this particular workflow will perform random forest models and it will also perform PLSDA. But the way we do that to make it a lot more robust is we perform repeated double cross validation, which means that whenever we perform a machine learning analysis, we do it 250 times and compare the results. This makes everything a lot more robust. It really reduces the chance of overfitting. However, it takes forever. So if you want to get this done quickly, I would suggest changing this option to no instead of yes. But if you have a lot more time and you want to see the machine learning analyses, you can always have this as yes. One thing to note here for the academics and the audience is this specific workflow is not available on GNPS2, but GNPS2 has a similar workflow that's called the Hitchhiker's Guide to Statistics, I believe. And you can access it in a very similar way that we just accessed this workflow. So again, once you have all of your parameters set how you would like it, you can always click the Submit Workflow button here. For today, again, we've already done that for you. So we're going to go to our tasks and we're going to find that statistical analysis workflow. When you view the task, you'll see you have the very similar options to what you had before. We have a lot of different summary tables for you to look at. We have some of the ability to get a table that gives you the presence and absence of each metabolite in each of your groups. You're always welcome to click through these at your own time after the workshop. Today, we're just going to check out some of the cool visualizations that we have integrated into this workflow. So the first of these is this visualized volcano plot right here. I have it already loaded. It might take a little while for you guys to load it. When it's loaded, you will have to choose which samples you want to compare. So in this case, we only have one option for each of these. We only have omnivore and vegan. But if you say had like 6 groups, you could choose which groups you wanted to compare using these drop-down menus. You'll also notice that we have a couple of different normalization methods that we apply to your data. So RCLR and median. And we also have the ability to look at your FDR adjusted p-values or your raw p-values here. Now, if you have questions about what those mean, again, I would highly recommend you check out the workflow documentation, which you can get to using that link there, or on the workflow input page, there's also a link for it. It will give you a lot more information about what those different normalization methods are, why we decided to use them. Also, if you say have your own statistical processes that you like to run, but you want to get a cleaned, blank subtracted, normalized data set to run those data on, what you can always do is you can click this Browse All Results button here, go to our feature tables, and you can just download the normalized tables that we use to run all of these analyses. So that's a really easy way to get started on your own statistical analyses if you don't like the ones that we're doing. So on the volcano plot, you can see we have our comparison of fold change versus p-value. If you have any specific metabolites you want to look at, like say we want to look at that subnetwork with all the bile acids, we can actually just filter this table here to look at that specific subnetwork. So if you click on these little lines here, you can filter the component, which component is another word for subnetwork. I apologize for the confusing terminology. You can put 13 in here, and you will just filter for the subnetwork that contains your bile acids. If you want to highlight any of these, you can just click on the row and it will highlight that metabolite on the volcano plot and also draw a box plot for that metabolite as well. You can also always just click on any of these to highlight it and draw a box plot for it too. And it will pin that metabolite to the top here so you can get more information about whether or not it has a library match, as well as what's its log fold change, etc. Also, just a side note, all of the compounds with library matches are either dark blue or dark red, while those that are unknowns are light red or light blue. So that's our volcano plot dashboard. We also have the ability to visualize a heat map, which you can check out by clicking on this button here. This heat map does clustering without knowledge of the input groups. So here it's nice to see that it clustered all of the omnivore samples together and all of the vegan samples together. But in general, for untargeted metabolomics, the heat maps look pretty. I'm not sure how useful they are because there's so many lines here, it's very hard to read anything. But you do have that option if you want it. We also have the ability to visualize PCA plots. So if you click on this button here, it will pull up the principal components analysis dashboard. You can see we have our omnivore samples on one side and our vegan samples on the other side, which means that these two groups are very different, which is nice to see. You can visualize up to 5 principal components components by just changing this slider here, and you can see all of the different PCs compared to each other. You can also see the variance explained here, and you can choose to visualize any of the 5 different principal components using these drop downs here on this menu. So if you want to create your own custom biplots, you can also do that. So again, I'm going to select our subnetwork of interest, which is 13 here. And I'm going to click on any of these to draw a biplot, which is just a line that shows you which group that metabolite is most associated with. We can actually look at all of these by hitting Control and clicking on a bunch of them. And we'll draw a biplot for all of the metabolites in that particular subnetwork. So that's our PCA plot. The last one is our machine learning models. You'll notice I did not run machine learning models here. So if you want to see what parameters I used for this analysis, you can always scroll down to your workflow task parameters, and you'll see I did not run machine learning. I was trying to run this quickly. I didn't have 2 hours to spare. So we don't have our machine learning models on this particular workflow. If you have questions about how to interpret them, I did just write a blog post that's up on our website that shows you how to interpret them. I also have one of those blog posts for PCA. So if you are not super familiar with principal components analysis, I'd highly recommend you check out that blog post as well, because it takes you through, what does each of these axes mean? What does it mean if things are separated versus not? So that's a really useful tool if you're just getting started with statistical analysis. So from this, this will give us a lot of really good information about which of the metabolites in our subnetwork are differentially regulated between omnivores and vegans. However, we still don't know exactly what kind of bile acids they are. And so we actually have a couple more tools that you can use to dig even deeper and figure out not just is this most likely a bile acid, but what kind of bile acid it is. And so to do that, we're going to go back to our tasks, and we're going to check out this MassQL task. Now, MassQL is a programmatic language for searching mass spectrometry data. I actually have a couple slides about how it works. So let's go to those. So MassQL is a way of saying, you as mass spectrometrists, you probably have a bunch of MS2 peaks or neutral losses or all that kind of stuff that if you see this, you know exactly like, oh, I see that peak, I know it's this, or I see this neutral loss, I know it's this. However, if you want to look through all of your MS2 spectra individually and look for those, have fun. It's like 10,000. Or you can use MassQL to look for them for you in 1 minute, which is really awesome. So the first thing you need to do in a MassQL query is tell it to look for MS1 or MS2 data. Here, most of the time, we're going to be looking for MS2 data. So you're going to go query scan info MS2 data where. Then you're going to add in your search parameters. So this search parameter is looking for a specific MS2 peak at 339.27. You can also look for all sorts of things. So you can look for MS1 data, you can look for neutral losses in your MS2, You can look for specific scan numbers if you're interested in that. You can also look for specific charges, retention times. It's a super flexible language. Now, once you have whatever peak or neutral loss you want to look at, you can then, oh yes, be careful using whole numbers on this. If you're using high resolution data, MassQL interprets 162 as 162.0000. So if you are looking for something that's 162.1, you might not see it. The last thing you're going to do is to choose your qualifiers. So this will tell the workflow what kind of mass tolerances you're expecting to see. So here we're looking for a MS2 peak at 339.27 with a mass tolerance of 0.01 m/z. But you can also add this in ppm, or you can tell it a percent intensity. So you say, I only want to see this MS2 peak if it's at least 5% of my total MS2 peaks. So this is a good way to say, get rid of those like noisy peaks that are like say 1%. You can also put in a specific value. So you only want peaks above say 100 or 1000. Only thing I would be very careful when you're using this is if you've used different mass spectrometers, you know that they all use completely different scales for how like intensity data works. So, if you're using a QE, it's like the lowest you'll get is 100. And if you're using a QTOF, it can be like 1. So just, if you're using data from different instruments, be careful that this intensity value is an intensity value that makes sense for the instrument you're using. So for today's MassQL query, we can go back here. You'll notice if you scroll down here, this is the query that we used to do this workflow. So here we looked for an MS2 product at 339.27 with a m/z tolerance of 0.01 and at least 5% of the total intensity. And we're also looking for a MS2 product at 321.26 with the same tolerance and intensity. So this is going to look through our entire molecular network and come back to us with all of the metabolites that have these two MS2 peaks in them. Now you can see the whole list of the metabolites that matched that particular search by clicking the query list here. But it's more fun to just visualize it in the browser. So you can click on Visualize Full Network Browse in Browser here. That will pull up the exact same molecular network that you were looking at previously. And if you go to node size right here, which is under Network Controls, and you click on it and click Enrich Query Index 0, this will change the size of your network nodes to match which of your metabolites matched that MassQL query. So actually, if you scroll in here, you can see that most of our metabolites that hit our query are actually in this same subnetwork that we've been looking at before. And what's really cool about this is that these specific MS2 peaks are only found in dihydroxylated bile acids. So now, instead of just, throwing these metabolites out because we don't have a library match for them, we now know that not only are they bile acids, they are dihydroxylated bile acids, and some of them are differentially regulated between people with omnivore and vegan diets. So that is what we have for today for the workshop part. As always, you are free to check out all of the different results that we have on this server, check out the documentation, get to know it a little bit better. But for now, I'm going to hand it over to Helena to talk about some of the cool things that the Center for Microbial Collaboration, Helena, you're going to have to help me out with that acronym again, is doing to help improve these library annotations and give a lot more publicly available stuff for the community.
Okay, yes, thank you, Sydney, for giving us the opportunity to talk about CMMC, the Collaborative Microbial Metabolite Center that we are developing here as part of this Ometa workshop. I'm going to start sharing my screen. Can you see it? All good?
All good.
Okay, can you confirm? Okay, yeah, thanks. Okay, so yeah, for the sake of time, and we also want to have like some minutes for questions and discussions later on, I'm going to just give a brief overview of what the CMMC is and what is our motivation for creating it and how you can use that in your research. So as Sydney mentioned, when we have an untargeted metabolomics experiment, we can do spectral library searches against spectral databases such as the ones that are available in GNPS or in Ometa. And then we are going to have about 10 to 20% of metabolite annotation when we use these spectral libraries. But if we are specifically interested in microbial metabolites, this is a very challenging task to be able to really trace this information of these annotations back to which microbes are producing or modifying these compounds. So basically what you can do is, okay, I'm going to get all the spectra matches that we get from the libraries, and I'm going to do a bunch of literature searches to see if these metabolites have already been reported to be produced or modified by microbes. And you can, I'm pretty sure that many of you here have already done that if you work with microbial metabolites. And this is definitely not something that is, it's something that is very time consuming. So in order to fill this gap, we on the CMMC, we are developing workflows to make the annotation of microbial metabolites more straightforward. So we have developed workflows in GNPS2. Currently, they are only available in GNPS2, in which people can upload information about microbial metabolites into a knowledge base that we are building. And we are also performing downstream analysis with the molecular networks as well, in which we can, for instance, color our nodes based on based on the reports of microbial metabolites that have been annotated in your searches. So we are building the CMMC with the community engagement. This is a center that is funded by NIH. So we are engaging with the community, and also with some other grants that are funded by NIH are also collaborating with the center. And we are providing several trainings, extensive documentation, and also office hours for people that are willing to contribute to these efforts. So these are some very current numbers from this Monday. So currently the knowledge base, the CMMC knowledge base comprises 29,000 MSMS spectra of about 1,800 compounds that have already been linked to 2,400 microbes. And for now, we have 30 people that have contributed to this effort. So basically, when you use this knowledge base that we are creating, as I mentioned, what you can do is a network enrichment analysis as a downstream analysis from your molecular networking jobs. And then what you will be able to see is, for instance, your networks that will be colored by the microbial metabolites that have already been deposited in the knowledge base. So again, here is another example for microbial bile acids that are available on the knowledge base. So these are all bile acids that are conjugated with amino acids and have already been proved to be produced by microbes. And then basically, when we have this network and this visualization, it's very straightforward. We can just click here on these nodes, and then we will know that this is cholic acid conjugated with serine, that it has already been traced to be produced by, that has already been traced to be microbial metabolism of a host-derived molecules. And then you can also see all the list of the microbes that have already been shown to produce these metabolites. And if activity is also available, you will also be able to readily see this information if it is deposited as part of the knowledge base. Another workflow that we developed as part of the CMMC is MicroBlast. So basically, as very, very brief overview, if we have a spectrum of a tandem mass spectrometry spectrum, what we can do is search if this spectrum was already observed in microbial monocultures before. So here, for instance, is an example also of a microbially conjugated bile acid. And then we can see here a taxonomic tree of which are the microbial monocultures that this specific this specific spectrum has already been detected. And what is very nice is that you can also search for unknown compounds. So for instance, you have an MSMS spectrum of an unknown molecule. You can also search that to see in which microbes it has already been detected. Well, here was a very, very brief overview for the sake of time, but we have, as part of the CMMC, already organized workshops. So we have a workshop for molecular networking, but we also have a workshop for micromass in which we go into more details on how you can use it, and also for the CMMC knowledge base and the network enrichment analysis. So these are the links in case you want to check that out. And here is also the link for the CMMC website where you also have more information about documentation and links to the workflows as well. So yeah, Sydney, I'm going to head it back to you.
Lovely, thanks, Helena. If you wouldn't mind pasting those links in the chat too, so that people can get to them, that would be awesome.
Yeah, I'll do that.
I'm going to share my screen again. So yes, so this is the end of the kind of formal portion of the workshop, but now we would like to open it up to questions. If you have questions, you can put them in the Q&A box, or you can raise your hand and we can let you speak. If you have questions after this workshop, I know I always remember my questions like the minute after the workshop ends. You can always check out the GNPS2 office hours if you are an academic. I'll paste the link for that right now. And if you are a commercial client, you can always feel free to email me with questions at my email here.